Hello, my name is Bradley Thompson, and I'm joined today with Mason Blocker and Bo Richards, and today we are presenting our project on leafy green production in the hydroponic system. And the hydroponic system that we were given was deep water culture. What is controlled environment agriculture? Controlled environment agriculture is a type of agricultural practice where through the use of technology, you're able to control the different environmental variables in order to optimize plant production. One of the goals of controlled environment agriculture is to reduce the variability among plants that are grown in other traditional agricultural practices like field production. Another goal of controlled environment agriculture is to optimize plant production so we can maximize our profit. One of the main benefits of controlled environment agriculture is that because we get to manipulate the environmental variables, we are able to tweak those variables to a plant's desired conditions in order to optimize plant production. The different variables that we can control is the amount of light a plants get, the temperature of the environment, the pH of the substrate or water, and the different nutrients. Controlled environment agriculture has many subsets, but the practice that we are focusing on is hydroponic production. And there are different hydroponic setups, but the hydroponic setup that we got was deep water culture. And deep water culture is basically where we grow the plants directly in the water. This setup is very low maintenance and low tech. It has a minimal risk of equipment failure like lines clogging, and the tubs are really easy to refill when water level drops. The main objective of our experiment was to analyze how different fertilizer concentrations affect the growth of different leafy greens. So basically, how we set up our experiment was we had 8 deep water culture tubs that were 4 feet by 4 feet and 6 inches deep. We filled up these tubs with 45 gallons of water and split them into 2 groups of 4. Each group represented a different fertilizer concentration. The fertilizers that we used for this experiment was Jack's 15-5-20 fertilizer. And for our first trial, the concentration that we used was 375 parts per million and 750 parts per million. The reason that we used this type of concentration in this trial was due to a mistake in our fertilizer calculation. So the concentration that we thought we were using was 5 times higher. In our second trial, we used 75 parts per million and 150 parts per million. So in each group, we dissolved its respective amount of fertilizer in each, and we placed these foam boards on top that held 36 plants, and the plants that we used were arugula, broccoli, lettuce, and spinach. Each tub had 2 aeration stones in each corner in order to maintain dissolved oxygen levels. And the variables that we measured was the height and width, the pH, the electrical conductivity, the temperature, the dissolved oxygen, the anthocyanin, the chlorophyll, and the plant tissue weight. The first chart I'm going to go over is the water added chart. So as you can see in trial 2, the amount of water that the plants used was significantly higher than the amount of water the plants used in the first trial. And that is because in the second trial, it happened in the spring and summer months where the temperatures were warmer, and therefore when temperatures are higher, the plant transpires more and evaporation is at its highest. Now I'm going to turn it over to Bo to go over the next few charts in our experiment.
So this is the graph part of the presentation where I will be presenting a lot of graphs that have a lot of colored bars in it. So I hope you are ready for that. The first one is anthocyanin. And as you can see, it's a general trend upwards throughout the weeks. Arugula, broccoli, lettuce, spinach all show a positive trend. And for each of them, usually it's actually the lower concentrations that did better, which is interesting. For chlorophyll, it's another higher trend upwards. And like anthocyanin, as the plant grows, you would expect it to have more in its leaves. For this one, you can see the 150 ppm did particularly well, especially in lettuce and arugula, while broccoli 375 ppm did the best, and spinach was a tie around which concentration was better. For temperature, it's again another gradual increase because we were growing crops throughout um throughout the season, it got warmer towards the end. So that's why weeks 3 and 4 for 75 and 150 ppm have the most, because it was just hotter during those times. pH is honestly one of the weirdest graphs in our presentation. The lower concentrations have a higher pH, and for arugula, it trends downward. Broccoli is kind of stable. Spinach takes a dip and comes back up. Lettuce has a strong pH. All of them go down to about the same results, and then 75 shoots back up. For EC is the easiest one because it's just measuring nutrients, and you can clearly see that the concentrations of the highest nitrogen have the highest EC, which makes sense. So you see 750 having the most throughout all of them, while 75 has the least. And so dissolved oxygen is another linear trend going downwards. And this makes sense because the air stones were quite small and could only pump the same amount of oxygen throughout the whole weeks. So as the plants grew and grew, they needed more oxygen. That wasn't being replenished as fast as they were growing. So that's why weeks 3 and 4, you tend to have low dissolved oxygen. So for arugula average height and weight, this is the measurements of how tall and how wide they were. You can see it's a general trend upwards as the plant grows, which would make sense, as when they grow, they get bigger. And you can also see, if you compare directly the arugula that 375 to 75 ppm, the higher concentrations did better than the ones that were at lower concentrations. For broccoli, it's much the same way. You have a trend upwards as the weeks go on. The plant grows higher, and this is pretty even between height and weight. And again, the concentrations with higher nutrients and have a higher fertilizer concentration grew more, which is, spoiler, the common trend between all the graphs. And so as you see the lettuce, it grew more over time, like the other two and like the next one, and the fertilizers did better, or the plant did better under higher fertilizer concentrations than the lower concentrations. The last plant, spinach, did the same thing. Grew more overall. The ones that had higher concentrations did better. Though in spinach, it's close. Spinach was the closest one between the lower ppm and the higher ppm. Now the harvest weight is just the weight at harvest. This is discluding the root systems of plants because they got tangled together and we couldn't identify which roots went to which roots, and we were only weighing a select few. So as you can see, arugula did not weigh as much as broccoli or lettuce. And it's actually interesting because lettuce is pretty even as with arugula. However, broccoli and spinach have a high disparity between different nutrients. 750 ppm has the most in broccoli and second most for spinach. And while you would think that 375 ppm does second best, but actually that's not true. And 150 ppm has the second best for spinach and broccoli. And I'll give it off to Mason to finish the presentation.
Over the course of our experiment, we tested 4 different plant varieties against 4 different fertilizer treatments. And we found that the fertilizer concentrations did impact plant growth, but we can't know for sure because of the many different dependent variables that we had going on environmentally, such as temperature, hours of light, which changed throughout the season, as well as like pH and dissolved oxygen. Pests were also a big issue for the 150 parts per million and 75 parts per million treatments, because those were done in weeks that were a lot warmer than the other higher parts per million treatments. And the pests really impacted root growth as well. Pictured here is the fungus fly, which would hatch larvae in the root systems, which caused chaos with the arugula plants and the spinach plants. The spinach plants actually ended up bolting because of stress because of this. And this is the red mite, which didn't really have a big impact at all, to be honest. So, continuing on, the table below on the bottom of the screen shows nutrient activity plotted against plant growth, which shows that as you increase nutrients, plant growth continues and rises, and then you reach a like critical zone, which shows like the acceptable range of nutrients. But no matter how much you increase nutrient concentration, it's not going to continue to grow more. You know, there's a limit. You reach a ceiling essentially, and then eventually, if you keep adding nutrients, it's going to hit a toxic zone, and that could be really difficult and harmful to your plants, same as a deficient zone. This table to the right of the screen shows the different nutrient concentrations that we used throughout our experiment, the 750 all the way down to the 75 parts per million treatments. And as you can see, with the higher concentrations, there's a lot more nutrients that are in surplus and definite excess, such as copper and zinc right here, which is, I mean, copper is over 3,000 and zinc is over 1,000% higher than what it should be. And then same with the 375 parts per million treatment, major surplus. And then once you get down to the 150 and the 75 parts per million treatment, you start getting into multiple nutrients that are deficient, and there's not enough. And then with the 75 parts per million, I mean, it's pretty much all red. It's in the negatives for almost every single nutrient. There isn't even enough nitrogen for the plants to reach their maximum growth. In the middle of each graph is the target amount, which through research has been shown to be the preferred target amount for leafy green vegetables in hydroponics. And in every single graph, the sulfur amount is 0 because the fertilizer we used did not provide any sulfur. This table right here shows what a fertilizer blend might look like when meeting the optimal ranges of nutrients for leafy green vegetables in a hydroponic system that is 170.34 liters, which was the volume of the tubs we used in our experiment. And this is what a blend would look like if it meets these ranges, and the ranges here meet all of the nutrients that are required for proper growth for leafy greens. So in conclusion, I would suggest changing this experiment in the future, or redoing this experiment and making sure that all the nutrients are at least met throughout the treatment. Because with the 75 parts per million treatment, as you could see in the previous slide, the arugula plants had yellowing leaves, which could be signs of nutrient deficiencies. And the 750 parts per million concentration, actually, we didn't see many signs of nutrient toxicity, but they did bolt earlier, such as, the lettuce, but the spinach looked great. So the optimal range for these plants could actually be higher, specifically for nitrogen and other nutrients as well. So further research needs to be done to show if, like that graph showed, if the parts per million treatments actually were in the acceptable range, or if they were kind of reaching the toxic range, but we can't know for sure. We would also suggest to control any dependent variables such as light, temperature, pH, dissolved oxygen, any environmental condition that can cause variation throughout the experiment. Because of these, environmental conditions, we can't say for sure if the disparities between each treatment is solely due to nutrient concentration. It's really important to do experiments such as this, especially in the controlled environmental agricultural industry, so that you know what you're using in your hydroponic setup or any kind of greenhouse setup is optimal for the most cost reward. without wasting any resources, because fertilizers aren't cheap and the world has a limited supply of them as well. So research such as this is, it's never ending, and it's important, because the more people you have on our planet, the more food we're going to need, and the more food is going to cost. So this research, is life-saving.